Hello my dear students, in the revision of biology question 2. Mention one function for each of the following: glenoid cavity, perianth, DNA satellite. We will form a table. In the table, the structure and the function. In the case of the glenoid cavity, the glenoid cavity is a cavity that located in the scapula bone, where the head of the humerus fit inside it to form the shoulder joint. But the perianth, we understand that the outer two floral whorls is the calyx and the corolla in the dicot flower. But in the case of the monocot, the calyx and the corolla has the same structure, the same color, and the same function. So in the monocot flower, the two outer, or the outer layer and the outer floral whorl, and the next one is called the perianth. The perianth protect the reproductive organs at the center of the flower against the presence of water, the wind, the dryness, and the other environmental factors. Number C, DNA satellite. DNA satellite is the two terminal parts of the chromosomes that protect or keep the structure of DNA against the action of the enzymes. So the presence of two parts of DNA, or the DNA satellite, which act as a two terminal part of the chromosome that protect the DNA against the action of enzymes, and they act as a non-codon parts, and the non-codon parts that protects the DNA and also may act as parts that to determine the codon parts of the DNA. The next question: illustrate by drawing only. The apparatus that used in the experiment show the ascent of water through the plant by transpiration pole. We understand that transpiration pole is a pulling of water from the root to reach the leaf on the higher trees. And conclusion only of this experiment. That means you should draw the experiment or the apparatus and put the labels on the diagram, and also explain the observation and the conclusion. We understand that in the diagram or the experiment, the presence of trough containing mercury and the tube with two opening. The tube is full of water, and cut the twig under the water surface to protect or to keep the water column inside the xylem vessels, and put the twig on the tube and close firmly around the twig or around the plant. Put the plant in a certain place or in the presence of air and wind, that suitable place that to facilitate the transpiration process in plant. We observe that the mercury level will increase or raises inside the tube because the plant transpire and causing pull of the water, and that causing a pull of the mercury that to replace the water which get out during the transpiration. This experiment to prove the presence of transpiration pull. We explain that the water can move or can ascend from the root to the leaf of the higher trees by the cohesive, adhesive, and the transpiration pull. In this case, the presence of the transpiration pull that help in the pulling of mercury upwards inside the tube. The next question: write a short note about the enzyme that participate in the process of DNA replication. We understand that the replication of DNA needs the presence of three enzymes: DNA helicase, DNA polymerase, and DNA ligase. The DNA helicase responsible for breaking down the hydrogen bonds between the nitrogen spaces that to separate the two strands of the DNA to help the duplication of the DNA. That means to help each of the strand act as a template to form the other one. So the helicase enzyme act as a direction from 3' to 5' in one strand, and from 5' to 3' of the other one. But the next enzyme is the DNA polymerase. The DNA polymerase enzyme that adds the nucleotides one by one on the direction from 5' to 3', or add nucleotide to the 3 end of one that to form one of the two strands, and add nucleotides to the other strand or to the 3 end prime. But the third enzyme, which is called the ligase enzyme or DNA ligase, the DNA ligases that are responsible for joining the nucleotides of the DNA that to form the other strand that act as a direction from 5' to 3', which act as the opposite direction of each of the DNA helicase and the DNA polymerase. That's the three enzymes responsible for the duplication of the DNA: DNA helicase, DNA polymerase, and the DNA ligase. The next question: what is the fate of each of the following after the occurrence of the fertilization process in the plant? The ovule and the egg cell. We understand that the ovule is the embryo sac which containing all the cells and the nuclei inside it, and the egg cell is one cell that located inside the embryo sac near or facing the micropyle. We understand that during the double fertilization or the fertilization of the embryo sac, we understand that the pollen grain containing two ovules, containing two generative nuclei. The two generative nuclei, one of them responsible for the fertilization of the egg cell into zygote, and the zygote can grow and change into embryo. But the other generative nucleus fused with the two embryo sac nuclei to form 3N. The 3N is the tissue that is responsible for the formation of the endosperm, and the endosperm act as a food stored to feed the embryo to help the embryo to grow. But after the fertilization, the egg cell is fertilized to form the zygote, and the zygote change into embryo. But the ovule fertilization that form the embryo and also the endosperm. May the endosperm is consumed by the embryo, and this in this case the seed is called the X-endospermic seeds, but the endospermic seeds containing endosperm as that in the monocot. But after the fertilization, the floral whorls as a calyx, corolla, stamens, and the other floral whorls fall down, but the ovary only will grow to form fruit, and the fruit containing the seeds and the two integuments that surround the embryo sac change into testa surround the seed. That in the case of the dicot plants, but in the case of the monocot plant, all the layers around the embryo sac fused to form grain, which act as a fruit with one seeds fused together. The next question. Question 6. The opposite diagram represents the ovule just before the fertilization. Mention the number and the name of the structure which, number 1, that is the diagram of the ovule that before the fertilization. So the first question is produced that asks about the structure that's produced from the mitotic division of the spore mother cell. One understands that the spore mother cell divided by meiosis to produce the embryo sac. So the structure that's responsible for that produced from the meiosis division of the spore mother cell is number 7, which is the embryo sac. But number 2 will fuse with the male nucleus to produce zygote. That's the number 3 egg cell, which is a cell at the center or surrounded by the two synergid cells and that facing the micropyle. The third part is the pollen tube enter, or the structure where the pollen tube enter through it. It is the micropyle number 12, and the micropyle is very important for the passage of the pollen tube for the fertilization of the ovule, and also responsible for the passage of water for the germination of the seeds. The next question. Explain each statement of the following. Number 1, the effect of the parasympathetic nervous system increased during eating food. We understand that during the critical situation, the sympathetic nervous system decreases the secretion or inhibits the secretion of the digestive enzymes, and also inhibit the contraction and relaxation of the alimentary canal, which is called the peristalsis. But the parasympathetic nervous system acts as the opposite action of the sympathetic nervous system. the parasympathetic nervous system, increase a series of contraction and relaxation or the peristalsis that mixes the food with the digestive enzymes, or increase the area of the food that exposed to the digestive enzymes, and also increase the secretion of the digestive enzymes to help in the digestion process. The next question. Explain each statement. The cholinesterase enzyme act on the returning of the nerve fibers membrane to its resting state. We understand that the postsynaptic membrane containing the acetylcholine, the cholinesterase enzyme to breaking down the acetylcholine after performing its function. We understand that when the nerve membrane is stimulated, or the stimulation of the nerve membrane help the passage of the nerve impulses in the form of polarization, depolarization, and repolarization, which act as action potential through the nerve fiber or the nerve membrane. But at the cleft, in the synaptic vesicles, or the synaptic vesicles containing neurochemical transmitter to allow the passage of the nerve impulses through the cleft in the form of neurochemical transmitter. If the neurochemical transmitter as acetylcholine reaches the postsynaptic membrane, that it changes its permeability, allows the sodium ion to move from outside to inside, and that it changes the state of polarization into depolarization. But if the cholinesterase enzyme breaking the acetylcholine after performing its function, that it changes selectively permeability of the postsynaptic membrane, and that it changes its permeability to allow the passage of the great number of potassium ions to move from inside to outside, to change the state of the nerve fiber in the form of the resting state, which is called the polarization, as is negative 70 millivolt. The next question. Two bottles containing 2 strains of certain plant seeds, both green. The 2 bottles containing green seeds, but one of them with the pure and the other with impure. Note that the green color is dominant over yellow. So the 2 bottles containing green seeds, and the green is the dominant character. But one of them is pure, but the other is impure. Explain on genetic basis how can you distinguish the genotype of each without using any other seeds. We understand that the green color is the dominant color, but the yellow is the recessive. So to determine the genotype of the dominant character, we carry out a test cross. But the test cross takes place by 2 ways. The first way is the crossing of the plant with the recessive individual, because the recessive individual pure, and that will produce the offspring, or the ratio of the offspring will indicate the genotype of the individual. If all the individual dominant, that means that the genotype is pure. But if the offspring produced 50-50, 50 dominant and 50 recessive, that indicates that the individual is impure. But in this question, that explains that by using these seeds only. That takes place by test cross, but the test cross in this case takes place by the self-pollination of both of them. If the self-pollination takes place in the case of the impure individual, C capital C small, that will produce offspring in the ratio of 3 dominant to 1 recessive. The recessive is yellow and act as a lethal gene. So in this case, we can't carry out test cross by recessive individual, because the recessive individual die. But if self-pollination takes place in the case of C capital C small and C capital C small, that's self-pollination of the same individual. In the case of the impure individual, that will form the offspring 3-1. But if the individual is pure, that means all the individuals produced are green or dominant. So the ratio of or the result of the self-pollination of the individuals, that will indicate the genotype of the individual. If all the individual will be green, that means the genotype of the individual is pure. But if the individual is impure, that will produce 3 dominant to 1 recessive. My dear students, that's the end of this part. Thank you very much. Goodbye and good luck.